Ever wondered why a tablet dissolves in your body and how it releases the medicine? Today, we unravel the mystery behind it. Dissolution, in the context of the pharma industry, is a crucial process that determines how effectively a drug will function in our bodies. It's the process by which a solid form of drug, like a tablet or capsule, dissolves in the body fluids to become a solution. The medicine then travels through our bloodstream, reaching the targeted parts of our bodies. Without dissolution, those little tablets would simply pass through our bodies, unused and ineffective. It's a complex process, influenced by various factors such as the composition and design of the drug, the conditions in our bodies, and even the way we ingest the drug. It's the first step in the journey of a medicine from the pharmacy to our bloodstream. So, in essence, dissolution is the process that enables a tablet to become medicine in your body. The journey of a tablet from your mouth to becoming an active drug involves some fascinating science. This process is called dissolution, and it's a critical step in drug delivery. Let's take a closer look at the basics of dissolution. Dissolution is the process by which a solid substance enters into a solution. In the world of pharmaceuticals, it's the step where a drug in solid form becomes dissolved in the body to exert its therapeutic effect. Think of it like a sugar cube dissolving in a cup of tea. The sugar cube represents the drug in solid form, while the tea is the solvent, typically body fluids like blood or gastric juice. Now, the rate at which the drug dissolves is called the dissolution rate. It's a crucial factor because it influences how quickly the drug will begin to work. A quicker dissolution rate means the drug is available for absorption into the bloodstream faster, which could be crucial for medicines that need to act quickly, like painkillers. But what determines this dissolution rate? This is where the Noyes-Whitney equation comes into play. This equation is a fundamental principle in the study of dissolution. It states that the dissolution rate is proportional to the surface area of the solid, and the difference in concentration of the drug in the solid and the solvent, and inversely proportional to the thickness of the boundary layer of solvent in contact with the solid. In simpler terms, the bigger the surface area of the drug, and the greater the difference in drug concentration between the solid and the solvent, the faster the drug will dissolve. Conversely, a thicker boundary layer of solvent can slow down the dissolution rate. So the speed at which a drug dissolves can be understood through the Noyes-Whitney equation, which forms the basis of dissolution studies. It's a fascinating interplay of physical properties and mathematical principles, all playing a part in how a simple tablet can bring about a therapeutic effect. How do we ensure that a tablet will dissolve properly in the body? Let's delve into the world of dissolution testing. Dissolution testing is a process of immense significance in the pharmaceutical industry. It is this very process that guarantees the consistency and reliability of oral solid dosage forms, such as tablets and capsules. When we talk about the efficacy of a drug, how quickly it dissolves in the body's fluids is a critical factor. Dissolution testing allows us to predict this behavior. A variety of dissolution apparatus exist for performing these tests. The most commonly used one is called the basket apparatus, also known as Apparatus 1. This apparatus consists of a small basket held by a motor-driven shaft, which rotates in a dissolution medium. Another widely used one is the paddle apparatus, or Apparatus 2. Here, a small paddle replaces the basket, stirring the medium at a constant speed. Other apparatus types, like the flow-through cell or reciprocating cylinder, are used for specific drug products or testing conditions. The process of performing a dissolution test is a carefully choreographed dance of precision. A sample of the drug product is placed in the dissolution apparatus. The motor is set to a specific speed, and the medium, often a buffer or hydrochloric acid to simulate stomach conditions, is heated to a specific temperature. The apparatus is then left to run for a predetermined time period. During this time, samples of the medium are periodically taken and analyzed to determine how much of the drug has dissolved. Dissolution testing is not just about ensuring the drug dissolves at the right pace. It also plays a vital role in the development of new pharmaceuticals, allowing scientists to understand how changes in the drug formulation affect its dissolution behavior. Dissolution testing, therefore, serves as a critical quality control measure in the pharmaceutical industry. It's a fascinating world, isn't it? one where science, precision, and a quest for quality come together to ensure the medicines we take are safe, effective, and consistent. What factors can influence the dissolution of a tablet? Let's explore. First off, let's consider the particle size. It's a significant player in the dissolution game. Smaller particles have a larger surface area relative to their volume, which can increase the rate at which a substance dissolves. Picture it like this. If you're trying to dissolve a sugar cube in your tea, breaking it into smaller pieces will make it dissolve faster. The same principle applies to pharmaceuticals. Next up is the temperature. Just like in our everyday experiences, such as making a cup of hot cocoa, higher temperatures can increase the rate of dissolution. This happens because the molecules in the solvent move faster at higher temperatures, which increases the frequency and energy of collisions between the solvent and the solute. Moving on, we have agitation. This is the process of stirring or shaking. Agitation helps to distribute the solute evenly throughout the solvent, which can speed up the dissolution process. Think about stirring your coffee to dissolve the sugar. It's the same concept. Lastly, we can't forget about the pH of the medium. The pH can influence the solubility of the drug and therefore its dissolution rate. Some drugs dissolve better in an acidic environment, while others prefer a more basic one. So the pH of the dissolution medium can greatly influence how quickly a drug is released from its dosage form. Each of these factors, the particle size, temperature, agitation, and pH, can be manipulated to optimize the dissolution process. But remember, it's a delicate balancing act. One factor can't be changed without considering its impact on the others. Understanding these factors allows pharmaceutical companies to optimize the dissolution process and ensure consistent drug release. This knowledge is crucial for developing reliable and effective pharmaceutical products. It's not just about creating a drug, it's about ensuring that drug can be effectively delivered to those who need it. While dissolution is a crucial process, it comes with its own set of challenges. Let's discuss some of these and their solutions. One of the primary challenges in dissolution is maintaining sink conditions. In the simplest terms, sink conditions refer to the state where the concentration of the drug in the medium is less than its saturation solubility. Ensuring sink conditions is critical to avoid reprecipitation of the drug after dissolution. This challenge can be addressed by increasing the volume of the dissolution medium or by using a substance that can form complexes with the drug, thereby increasing its solubility. Another challenge is related to hydrodynamics. The movement of the dissolution medium can significantly impact the dissolution rate. Factors like agitation speed, type of apparatus, and the shape and size of the dissolution vessel can all influence the hydrodynamics of the system. To overcome this, it's important to standardize these variables during dissolution testing. Then there's the issue of variability. Variability in dissolution testing can arise from a variety of sources, including equipment, operator, and method variability. This can lead to inconsistent and unreliable results. The solution here lies in strict adherence to standard operating procedures, regular equipment calibration, and rigorous training of operators to minimize human error. And let's not forget about the advances in technology that are helping to overcome these challenges. For instance, automated dissolution testing systems can reduce operator variability and increase efficiency. Similarly, the development of bio-relevant dissolution media, which mimic the conditions in the human body, can provide more accurate and predictive dissolution results. Indeed, dissolution is not without its challenges. However, with a clear understanding of these issues and the strategies to address them, it's possible to ensure reliable and accurate dissolution testing. Despite these challenges, the pharma industry continuously innovates to ensure effective dissolution and drug release. To sum it up, dissolution is a fascinating and essential process in the pharma world. This journey took us from the very concept of dissolution, a process that transforms solid dosage forms into a solution, paving the way for the body to absorb the medication. We delved into the science behind dissolution, understanding how it's not just a simple matter of dissolving a substance, but a complex interplay of different factors. Dissolution testing, a fundamental step in the pharmaceutical industry, ensures the quality, efficacy, and safety of the drugs we consume. We also explored the various factors that can influence dissolution rates, from the composition and design of the drug to the pH and temperature of the environment. Addressing the challenges in dissolution testing, we discussed potential solutions, emphasizing the importance of continuous research and development in this field. And that wraps up our journey through the dissolution chapter in the pharma industry. Remember, every time you take a tablet, there's a whole world of science that goes into making that medicine work for you.